Guys, so today we're going to discuss about the early embryonic development, all right? So previously we discussed about the fertilizations, right? Remember, the fertilizations takes place in ampulla regions, right? Now, the whole concept of the early embryonic development, what is happening in the first week? Look, as we said, the fertilization takes place in ampulla regions. But now, think about this, right? This is happening, within the 24 hours. Let's just say within 24 hours. As soon as when it penetrates the zona pellucida, right? The fertilization begins. The sperm penetrates the zona pellucida, that's where the fertilization begins, right? And this process takes place within the 24 hours, right? Then after that, what happens is that after 24 hours, right? Like basically from, let's say, day 1 to like 2 days, or sometimes we say like 30 hours too, right? In this day, what happens is that these cells, they go process of something called cleavage, right? And you have to remember what cleavage means. This is very, very important because cleavage basically means cellular division, to be like really easy. The process in which where the cells go through a whole process of G1, G2, right? Aspase, right? What is this? This is called interphase, right? And then you have a mitosis will occur. And then also you have a cytokinesis will occur, right? All these three phases, that will take place in every time when the cleavage happens, meaning the whole process of the cell cycles will occur here, right? And now, remember here, what is happening here is that this is happening in the first 24 hours, right? And then after that, in the second day, right, what happens is that these cells will go and divide into two, right? When they divide into two, right, this is zygote, right? This is zygote basically. So look at this. These cells divide into two, or divide into two daughter cells, right? That is basically called cleavage, and they went through cellular divisions, and now this zygote is in half, right? These cells, we can also call them a zygote, or we can also call them a blastomere, right? And this is happening around like day 1 to 2 days. That's what is happening, right? Now, remember, when the fertilizations, in this case, right, what happens in this case, when the fertilizations happen within the 24 hours, before this breaks down into, let's just say, blastomere or two zygote, right, two cell stage, the centromeres are sort of like, arranged or lined in basically longitudinal fashions, okay? And these are basically microfilament. And this is basically showing you a fertilization phase, all right? Now, this stage, which happens 1 to 2 days, this is a two-cell stage. We can simply write down here. And this is still happening in where? Fallopian tube, right? Now, then after that, again, what happens is that, the cells, like right here, these corona radiata cells, basically they started going through process of basically apoptosis, right? That's what happens here. And they lose apoptosis. They lose this corona radiata cells. But what they don't lose is this zona pellucida layer. And you have to remember, there's importance of zona pellucida, okay? Because it basically prevents these cells to actually get hatched, or basically hatched out of this layer, right? So, and basically it's preventing implantation to occur. The role of zona pellucida, right? This basically providing a covering coat, right? Protection layer to prevent implantation, like to prevent immature implantations, all right? Now, the two cell stages after what happens is they will still go further cellular divisions. They'll go through all of this phase, and they'll basically break down into, what is this called? 4-cell stage, right? This is a 4-cell stage right here, like this, right? 4-cell stage. This is what's happening here. Now, this 4-cell stage, okay, this is also happens like around, let's just say, you can simply say this around day 3. Right? So remember, day 1, you can say there's 2-cell stage happens. Maybe day 2, let's just say. Day 2, there's a 4-cell stage happens. And then after that, there is further division happens, right? And then they will go and divide it into, let's just say, 8-cell stage, right? Like this. 8-cell stage. And these 8 cells are, what do we call them? This is still the blastomere phase. And this is maybe happens in like day 3, right? Now, after day 3, this is, let's say, 8-cell stage, right? 8 cells there. But remember, what is the most important thing you have to remember here is that, as these cells are, going through process of cellular divisions, and after like a day 3 layers, day 3, and there's zona pellucida is again here, right? What really happens is that, like inside these inner cells, like these cells right here, what really happens is that around the day 3, right, after that, what happens, they start expressing like different genes, right? And that different gene, okay, induces, right, for the cells, inner cells, to get like, differentiated into some other stuff. But what is that happening? So basically, if I make it down here, let's just say this. Now, if I make it there right here, all right, and let me draw in black, right, like right here, and then you have cells right here. I'm going to make the cells, all right? Look, like this. Arrange stuff right here, okay? And what is this? They started making more compact, right? What cells are this? These layers of the cells, right, which are getting more impact and layer, because in up to 8 cell stage, these cells are loosely arranged, right? But then after day 3, right, what happens is that these cells are started coming together and start getting packed together, right? And they actually go in further cellular divisions or further cleavage and make something called 16 stage, okay? And then when you look at the 16A, there's a bunch of collections of cells out there, right? That cells, okay, we no longer call them, it's the blastomere, okay? What do we call them? At the 16 cell stage, like this one, let's just say, I think I have more than 16 cells right here, but maybe I have only 16, but just think about the 16 cell stage. You can simply say that this is 16 cell stage, right? This 8, the 16 cell stage, we call them a morula. And why we call them morula? Because it appears as mulberry, right? That's how the scientists found it. Now this is the important concept, right? The 16 cell stage is called morula, right? All the way up to here, 2 to 4 stages. This is a blastomere phase, right? Blast means like rapidly proliferating cells. I'm actually showing you how actually the nucleus looks like. And again, I got this image from the Langman. So if you look at it, there are 2 cells here with the jagged cleaves. It makes 2 cell stages, and if you look at this one, there's a 4 cell stage, and this is a morula. Now, again, look at this. This has happened day 0, right? We can simply say the fertilizations happen less than 24 hours, right? Or within the 24 hours. And this is a 2 cell stage. We can simply say this is day 1, right? Repeating this. This one, 4 stages, you can simply say this happens in day 2 days. Day 3, maybe 8 cell stage. And then day 4, this is where the 16 cell stage or morulas are formed, right? As I discussed, in here, during, like after day 3 or so, there are different sort of genes are expressed, right? And that basically causes the cells, like if I have to make this super clear, let me draw this. You have this cell right here, right? The sum of the cells, right? What happens is that they started like, proliferating and making the mass like this, They started making a compact layer, all right? They started making sort of like compact layer. And that is the process of where their, cells are arranging or started coming tight together because they're making a tight junction. That is called compactions, all right? That's what is also happening. And obviously, you still have the zona pellucida, right? And around the day 4, what happens is that, or after the day 4 or so, or maybe day 5, what happens is that this 16 cell stage, morula, that basically, sort of like, get launched, or maybe like, get sort of like comes into this state, and this is called uterine cavity right here, right? The uterine cavity here. This happens, let's just say in the day 5 right now. And then you still have what is this layer? You have a zona. You have the zona pellucida, and these cells, this 16 cell style, they started going more cellular divisions and making more layers of cells. And ultimately what happens is that, these cells are sort of like started arranging themselves into more, like this compact together and making mass of the layers like this, right? That's what is happening. And then what happens is that they start making a mass like this. Look. This is what happened, right? Now, they started coming together and they started getting packed together. And these cells, right, the inner cells, right, these cells, which is in the central, this cell, these are called central cells, right? These are called central cells, right? Are centrally located. And these black cells are what? Peripheral cells, like peripheral cells, right? What am I saying? What is the importance of this? Look, the centrals and peripheral cells. This central cells, right? This is going to form something called inner cell mass, right? And this periphery cells mass is what's going to form something called outer cell mass. Because this inner cell mass is the one that's going to make future what? Embryoblast. Your Langman says it's embryoblast, right? Embryoblast. This is why this is the importance of that. And then this outer cell mass is what's going to make a trophoblast. Tropho means like they're providing nutritions, right? That's what's going to make. And this trophoblast is the one that's going to give future rise to placenta, let's say. Remember, and how do you know that whether, how do these cells know, let's just say, how do these cells know that whether or not they're going to make inner cells or outer cells? It has to do with the different genes, all right? There are so many different genes out there, What happens, that get expressed and that basically causes or induces this middle or center portions of the cell to differentiate it into the inner cell mass, right? And then the outer layer, the peripheral layer, they'll go and make the outer cell mass, which is called trophoblast, right? Now you have to remember this terminology, all right? Now, and again, there's a zona pellucida is there. But as this floating in the uterine cavity, what happens is that, this zona pellucida are leaky, And what happens is that fluid started going, the fluid down here started accumulating, right? And on top of that, some of the cells right here, this started secreting like the fluid too. And basically that causes these cells, like let's just say this, okay, this, and this is again like this, so that's a day 5 here. And here, there is a mass, okay, there's a zona pellucida is there, and then look, there's a layers of the cells, right? And this is outer cell mass, it's a trophoblast, right? And then there is this cells right here, embryoblast. When the fluid started secreting, right? What happens is that fluid started secreting down here. They started forming this cavity, all right? They started forming this cavity. This cavity you get to see. And this, whenever there's a formation of cavity and this cavity contains this fluid, then we call them now we no longer call them a morula, okay? We call them a blastocele, or this cavity is called blastocele cavity, all right? This is called blastocele or blastocele cavity, all right? Remember, it is very important that when the fluid start accumulating and forming the cavity from the morula, right? So now that formations the cavity, and you have the layer separated, right? This outer cell mass, trophoblast, and then embryoblast is there, and there's a fluid cavity is there. This is called your blastocells, right? And these blastocells, right? This is the blastocells. It's going to get implanted, right? Now around, let's just say, now really what happens is that when the as fluid started accumulating and all, there's also what also happens is that there's also degradations and loss of this zona pellucida happens, right? This zona pellucida happens. And when there's a loss of this zona pellucida happens, what happens is that this cell, this blastocells, right? Remember this, I said blastocele, not blastomere. This is blastomere, stays. This is blastocele. These blastocells will get hatched out from this zona pellucida, and then it will go to the body. Remember, this uterus has what? Fundus. This is cervix, right? This is body. And basically what happens, the posterior superior surface of the, let's just say this endometrium, right? That's where this blastocells will get implanted. Let's just say right here, right? And then making the mass like this, right? And then you have cells. Look at this. Now, this is happening around day 7, to be very, very specific, but usually it will happen from like, you could say anywhere from like day 6, let's just say from anywhere from day 6 to 10, all right? But to be very, very precise, it's like day 7 days, right? And it's going to get implanted. The blastocell gets implanted. One of the functions, as I already said that, having the zona pellucida, it's basically preventing premature implantations, all right? Now, you have to remember that around this, like around day 6 or like around day 7, what happens is that this embryoblast is also, started secreting something like, let's say, fibroblast growth factor. I'm going to take these pictures, I'm going to put it down here, okay? Look, what is happening here? is that if I make this in a black right here, right, you have the cells like this, right? Look, this is what? Trophoblast, right? And then you have this, let's just say embryoblast right here, like this, right? Now this embryoblast, if I make this cells, you know, embryoblast or inner cell mass, right, which is going to give rise to intraembryonic tissues, and then this outside layer, which is going to give rise to extraembryonic tissues, you know, this other stuff. Remember, these cells, this will start secreting, like, for example, fibroblast growth factor 4, one of the proteins, right? And that induces these cells, right? These cells to basically what? Go a process of further divisions, and they started forming what? Layers of cells, like forming the cells called, what are the cells called? And then, let's just make it green. And then, look at this. They started forming something called bilaminar disc, right? And this bilaminar disc, right, when it's formed, there's obviously fluid cavities there. This is what it's called. Now, when they form a bilaminar disc, what do we call them? What do you call them? This embryoblast. This first layer, which appears as columnar, this is called epiblast. And the one that is more like a cuboidal, we call them a hypoblast. Okay, remember, this is usually takes place, the bilaminar disc usually takes place in what? In the second week, right? This is what happens. And remember, this epiblast is the one that is going to go and give rise to the 3 germ layers, like, for example, ectoderm, endoderm, and mesoderms. This is later that's going to go and give rise to ectoderms, endoderms, and mesoderms, right? So this is something that you have to remember. Bilaminar disc is taking place here. The most important concept that you have to remember here is that sometimes, you know, there's a premature implantation can occur, right? If, let's just say, what can cause premature implantation? One of the things is that, look, if there's no this zona pellucida is there, then that can cause premature implantations. On top of that, let's just say sometimes this implantation can occur around the cervix area, right? If this happens, then we call them out. That is a pathological conditions, right? Because that can cause hemorrhage. And we'll talk more than in pathology, but that can also happen. And other locations that can happen, right? And one more thing you have to remember here is that implantation not only happens on the ampulla, but other locations. Like, for example, here, down here, I could even make like ovary, right? And if it happens in ovary, then that is an ectopic pregnancy, right? So again, guys, this is a very, very important concept about what are the things that's happening in different cell stages, right? Day 1, let me quickly repeat this. Day 1, there is a zygote, gets the 2 cell stage happens. Day 2, 4, you know, day 3, 8, maybe 16 stage compaction just happens. And after that, maybe in day 6 or day 7, that's where the implantations will occur, right? And remember one more thing that you should remember here is that, you know, normally, look, if we make this, let's just say that if the female cycles, it's like 30 days. Let's just say this is 30 days. This is like a 1, and then let's just say 30 days. That means our ovulation should be around like 16 days, right? So what has happened? This is like a follicular phase, right? This is a menstrual cycle, and this is a secretory phase. That's what happened. And here, if you look at this, what is happening to estrogen level? It goes something like this, right? And then if you look at it, hold on, this is more like estrogens right here. Sorry, this is progesterone, right? And what happens here is that if this is, let's say, ovulations happens, if I make this as LH phase, like this, right? So if you look at this, this is, let's just say, because of the LH, this is LH surge, right? This is around 16 days. Look, but then what is her fertilization day? Fertilization day happens, if this is her fertilization, this is like on 16, this is her first fertilization day. So if you count all the way go, and maybe like around 6 days, what is happening? Or 7 days, implantations happen. Let's just say 7 days, the implantation happens, right? So this is like in the secretory phase, that's where the implantation is happening, right? And who is driving this implantation phase? This hormone. What is this hormone? Progesterone. And who is releasing the progesterone hormone? Remember, when those graphene follicles, they get rupture, right? They release secondary oocytes, but they also form temporary female endocrine gland, right? That is called corpus luteum. And corpus luteum, right? This corpus luteum, that secretes progesterone. And progesterone makes this endometrium, right? This, what the endometrium glandular, right? And also very, very vascular, right? Very vascular. That's what it makes. And they also makes this cervix, what? Thick, right? Cervix plug. That's what. So that way that the no sperm can actually travel through. That's what the progesterone do, right? And it basically makes this whole endometrium extremely vascular and glandular. So that way that, this implantation can occur smoothly. So that's what the progesterone do here, right? But to maintain, right? But usually the corpus luteum, they degenerates after 12 to 13 days, right? 12 to 13 days. That's what happens. But to prevent degenerations of the corpus luteum, really what happens is that this cell, this outer cell, this trophoblast, they also start differentiating into other cell types. They go and further break down into, basically, these cells will go and break down into, start making layers, because they're giving extra embryonic layer that's going to make cytotrophoblast and syncytiotrophoblasts. Okay, they'll make cytotrophoblast and then syncytiotrophoblast. And this syncytiotrophoblast releases something called BHCG, beta human chorionic gonadotropin hormone. And this beta BHCG, they go and communicate to the corpus luteum to continue producing progesterone for the product of conceptions. All right. So this is something that you have to remember. All right. I got this picture from the Langman, and I'm showing this to make your concept even more clear. So if you see the number 1 right here, right, this is actually the ruptured follicles, right? Once it's ruptured, what happens? Look at this. This is a corona radiata right here. If you look at this photo right here, the corona radiata are there, right? And then you have gena pellucida, and this is basically a secondary oocyte. This whole is a secondary oocyte, right? And then after that, if you actually go right here, this is where that you're talking about the fertilizations are going to take place right here, okay? And then after that, this is where the DNA replications start will happen. This is what this number 3 is showing. And if you look at the number 4, like remember, this is where the both paternal and maternal pronucleus are fused together and they form the zygote. And then basically in this right here, within 35 hours, the zygote goes something called split up, right? This is where the cleavage happens all the way up to here. This is how cleavage happens, right? And then around around this the photo, this is around happening at day 3 or day 4. What happens is this morula that actually gets sort of expelled out into the uterine cavity. And if you look at right here this photo, what is it showing? It's showing you that the cells are actually this showing right this picture right here. This one is showing you the inner mass, and this outside is basically this showing you, if I make it right here, this is showing you the outside of the outer mass, right? And around the day 6 or so, the implantations occur. That's what this image is showing. All right.